So my lab studies the effects of diabetes in the heart. So how does diabetes over time affect the calcium dynamics? So your heart is governed by contractions. So how does the contractions of the heart change depending on the progression of diabetes? So the idea is we see some proteins go down that affect calcium dynamics, and we're going to try to reinsert those proteins and see what it does. So we're talking 3 to 5 years down the road and stuff. The other project that we're focused on is the effects of GLP-1 agonists in the heart long term. So those GLP-1 agonist drugs are being used for type 2 diabetes and also for weight loss medication. So there are a lot of positive outcomes of it, that it's helping with contractions and also be arrhythmogenic. So we're trying to see what's going on with GLP-1 agonists long term. So the first year, it's a lot of hands-on training. It's a lot of, they come to the office and I show them how to do some software analysis. I try to do everything where I show them, because if I have other students show them, then it's like if the other student learned something wrong, then it goes to the next generation of students and then the next generation. So I do have other students teach, but then I go in and I also teach myself to see if everything is okay. Not only physically doing injections, doing routine breeding, routine weaning of mice, genotyping, ear notching. On top of that is the whole cascade of understanding how to plan your experiments. Like, are we doing 5 mice? How do we get 5 mice from our mating groups and things like that? So all of the mouse handling should be kind of targeted and completely hashed out by the first year. And some of the basic molecular biology techniques, Western qPCR, and some of our calcium dynamic techniques like confocal. So everything gets done along with doing like sample experiments, but also the analysis endpoint, plus all the animal handling and stuff. A lot of work. I think they're pretty comfortable. I do often tell my students, I think you're totally right. I think we should change this. And I like that. I like when students are not just receptive of, let's do it this way. It's more like, I looked and I found this paper that showed this other way to do it. That shows initiative and it shows independence and it shows like true curiosity. That sometimes when I look for things, I quickly glance at papers and stuff and I'm like, okay, I think let's try this, because there's a lot of other things to do and then there's other things we need to kind of hash out. But the fact the student went in and kind of took more time, I really favor that. I think they do push back, and I like that. I like that. More brains are better than one.